right? We're going to finish up the notes for chapter 14, which is nucleic acids. Now remember, we said DNA can make copies of itself. That's called replication. But if we need to use the DNA to make something, first thing we do is the DNA opens up and a copy of it is made, but it's not exactly the same because RNA is making a copy. So remember, A and U would go together and you have a different sugar and so on. But a part of this is going to open up whatever gene you need, and RNA is going to make a copy of it, and it's only going to be single strand, and that's called transcription. Okay, so transcription, is making a copy of part of the DNA, and then it's going to leave the nucleus, okay, and then it's going to go out. It's going to kind of attach to a ribosome, and it's got this code, and every 3 letters of a code is called a codon. Okay, so like AAA would be a codon, or CCC or CGG or something like that. Those are codons, and every codon has an amino acid that's attached to it. So the amino acids come up and they attach, and then as you get more and more amino acids, you have your protein. Because remember, proteins are a sequence of amino acids. Okay, so the sequence comes from the DNA, and it's the code, the nitrogen base code. Okay, so protein synthesis, again, it's 2 steps. First, it's transcription. Inside the nucleus, a part of the DNA opens up. Now, when you replicate DNA, when you make a copy of DNA, the whole thing has to open up because you need a copy of the whole thing. But if you only need a small piece of the DNA, that would be a gene, and it's copied by, again, messenger RNA. Okay, so it's a single strand, and it copies the information that's needed. Okay, that's called transcription. Then translation is taking that messenger RNA, it's going to attach to a ribosome, and the messenger RNA is like a template, okay? Its job is to assemble the amino acids, and again, the amino acids are going to come from, they're attached, every amino acid is attached to a transfer RNA. So what will happen is, let's say the code sitting out there is CCC, okay? Transfer RNA with GGG, and it'll bring its amino acids, and now on the transfer RNA, the GGG would be the anticodon. So the codon and the anticodon go together, and that's the order that the amino acids come up, whatever those nitrogen bases. And that's called translation. So transcription is making a copy of the DNA and through messenger RNA, and then translation is actually using that template, that messenger RNA, to make a protein. Okay? Transcription, when a protein is required, again, when you need to build a protein, there's a gene in your DNA that says, yep, this is the protein you need, so that part opens up. And then during transcription, again, that part, that gene, is going to open up, and that messenger RNA is going to go in there and make a copy of it. Now, it's just a single strand, okay? Replication is different. That's making a copy of the DNA. But when you just need to use a small piece of the DNA, excuse me, messenger RNA will come in and make a copy of it, okay? And then it'll close back up again, which is kind of cool if you think about it, okay? And again, it goes from the 5 to 3 side or the 3 to 5, depending on which side of the template opens up. Okay, so in that respect, it's a lot like replication, only now you're going to have 1 strand, and then you're going to have the differences of RNA. You're going to have the different sugar, you're going to have uracil instead of thymine, that kind of stuff, okay? And again, like, well, I just said that, uracil is a substitute for thymine. But it's the same kind of process. It's going to match up, okay? And then to do this, there's something called Rna Polymerase. It's another one of those enzymes, which is actually made from DNA. It's the one that starts that transcription. It opens it up, and so that you can do that. Okay, and then other than bacteria, the mRNA is like what they call pre-mRNA. It has to be cut and spliced before translation, so it has to be read and used. Okay, bacteria a lot of times don't have DNA. A lot of times they just have RNA. Okay, so translation now we've got this messenger RNA. And every 3 nucleotides, so every nitrogen base is, like I said, you have a codon. It's very specific. Every amino acid has a codon that goes with it. So I'll show you a chart of it, and this is in your book as well. But there's actually 64 possible codons they make up all of the genetic code. But now remember, whatever the codes are, you can double up. You can have two amino acids, same amino acids in a row. You have these 20 amino acids, and the order that you put them in determines your protein. It can be 50 of these, 50 amino acids, which would be 150 codons, or 150, I'm sorry, nitrogen bases, or it can be, 27,000. It just depends on the protein, okay? But the genetic code is the same in all organisms, and what it does is the beginning code is AUG. Okay, once it sees that, that's where it starts. So it can be reading the DNA and going along, and until it sees AUG, nothing happens. So it needs a start codon and it needs a stop codon. Okay, and here's the three that are stops. So in other words, protein's made, time to quit. Okay, because the messenger RNA might be longer than it needs to be. So it's not going to start adding amino acids until it gets to AUG. So the match of it would be UAC. UAC. And then when it runs into one of these guys, then it stops. So it's like, oh, we're done. We made the protein. So we actually had start and stop codons. And then we have codons. The remaining 61 codons are for the actual amino acids. Most of them have more than one. And again, I'll show you the chart of them. It's not just UUU is one of the amino acids. It might have more than, might have like 3 or 4. Okay. But generally the variation occurs in the third nucleotide. That's usually where it happens. And that's usually where you can get some errors. But again, they have all these enzymes that read stuff and fix stuff. It's amazing, that system that we have in our cells that takes care of us without us ever even realizing it. Okay. So mRNA is coded into a protein, but a protein cannot be coded back into the mRNA. Kind of weird, right? So it always goes in one direction. It always goes from the DNA to the RNA to the protein. It can't be the protein makes mRNA and that goes back into the DNA. It doesn't go backwards. It only goes in one direction. Okay. Here's what I was telling you about. So like for phenylalanine, I think it is these are the two codes for it. UUU, UUC. Okay. Leucine actually has these two and these. And the way you read this is it's like, here's the first one, C. Here's the second one. Here's the third one. So like CUU is where they got this code. But anyway, these are your different amino acids. And you can see they all have, like argon has, it's arginine or something like that. Anyway, it has these 4 and these 2. So they have more than one code that codes for them. But again, this amino acids attached to a piece of RNA that has this code on it. Okay, so it looks for that code and then it'll drop its amino acid right there. Okay, and again, the amino acid is what determines what protein you have. Okay, so again, translation occurs at the ribosomes. And the ribosome has like 2 subunits. One has a little piece of ribosomal RNA, and then there's an active site. So what'll happen is the RNA goes through, it's kind of like a teletyper, it goes tick, 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 tick. And as it goes ticking through, then it adds the amino acids. And so the RNA is like the thing that forces it through, and then they add as they go through it. So it's better if you watch a video of it. Me explaining it is probably confusing you. But basically, the amino acids are added at the ribosome. Okay, so during the protein synthesis, they lock around that messenger RNA. And again, they go from the 5 to the 3. They read one codon at a time. So you have a codon matched with an anticodon. And it's just this code, like I said. If the codon is CCC, then it's going to look for an anticodon that's GGG. They're just going to be the opposite of each other. Or they're going to be the ones that bond together. Okay. If you assemble your amino acids, that's what the transfer RNA does. And I'll show you, this is probably better. This shows you what transfer RNA is. So the amino acid is up on this end. And this is where the anticodon is. So if this is messenger RNA, and you can see it's GCC, this is the codon. Okay? So it's going to look for the anticodon on a transfer RNA that has, so G is going to look for C, G, so here's GCC, this is going to be CGG. They match up, okay, and it brings its amino acid. So alanine is the amino acid that goes with CGG or whatever, okay, And then now there'll be another 3-letter here. And so this will drop off its amino acid and then it'll take off. And then another one will come in and hit this code. Okay. Whatever that is, it'll bring down its amino acids. And then the amino acids just kind of join together, join together, join together. And they're joined together by a peptide bond. Okay. So they just keep, this is the order that they go in, is whatever the messenger RNA is. And it's every 3 letters, that's a code. So first you have to start with AUG, that's the start, and then it adds and adds and adds and adds and adds, and then eventually the amino acids will get to a stop codon, they'll all join together, and you got yourself a protein. Okay? Okay, so during translation, the base pairs, they're going to look for a codon for the messenger RNA and an anticodon from the transfer RNA. They're going to match up, okay? And they call it the wobble hypothesis that allows the non-standard pairings in the 3rd position. Not a big deal, you guys. You don't have to know that. But again, there's all kinds of things about that. But basically, it's matching up their code. Okay, the base pairings between the anticodon and the codon bring the amino acid to the ribosome, and then they start forming peptide bonds between them, and then you got yourself a protein. Okay. The transfer RNA will drop off its amino acid, but it's got everything else still on it, so then it'll go back and pick up another amino acid so it can be recycled. Okay, and again, your polypeptide chain is your protein. Remember, the primary function is the order they're in. That's, whatever the amino acid is, that's your primary. And then they'll start doing their secondary, which is either they'll fold or they'll spiral, helical or folded. Okay, and here, I hope this explains it. Here's our messenger RNA and its little codes. Again, here's my start codon, brings it in. So then this one's going, oh, I belong there because there's GGG. I'm going to bring CCC here. And this one started, this was the first one, so it adds on to it. And then we bring another one and it adds on to it. And you just keep adding them and adding them and adding them. And they go in the order of whatever they match up to. And then they'll go until they hit a stop codon. And then it'll release and then you got yourself a protein. So that's how you make a protein. This comes from the DNA. And then the transfer RNA bring their amino acids. These letters are the codons. These are the anticodons. And they match up and they bring their amino acids. Okay? So a genetic mutation, if you change one of those codes, if you think about it, if you change this, let's say this one gets lost and then you move all this one down. Well, now instead of GGG, it's GGA. That's a different amino acid. And then this one's going to be messed up and this one's going to be. So if you just lose one of those, you're going to have some problems because you're not getting the amino acids in the right sequence and then you're not getting the protein. Okay. So that's a genetic mutation. Okay. You can be born with, you can be born with that. It can be something that happened already and it was passed on to you, or it can actually happen in your chromosomes, depending on. Okay. It can happen because of, being exposed to chemicals, things like that, cigarette smoke, all that kind of stuff. So if one nucleotide is substituted for another, that's called substitution. And again, you can have some problems. Sometimes it doesn't matter because remember, 98% of your genes or your DNA doesn't code for anything. So if it's in that part, you're golden. You can get a frame shift where one nucleotide, like I said, is added or deleted, and then you're going to have all the way down is going to be messed up. That's an issue. That's going to be a problem. Okay. Both types of mutations can change the amino acid sequence. That means you're building a protein that maybe you don't need, or you're not getting the one that you need. Okay. And here is, again, normal gene sequences. Again, if I throw in another one here, then that throws off this, that throws off this. This is what it's supposed to be, but now it's not that anymore. This one is in the wrong order. Or if you get a shift one where the whole thing, again, the first two are right, but then everyone after that is wrong. And you're going to have some, there's going to be some severe consequences because of that, because you're not going to get the proteins that you need. Okay. So I'm trying to get done here. Okay. So again, if you have DNA mutations that can be minimized by the redundancy, like I said, a lot of the DNA doesn't code for anything. So, and sometimes if you get a codon of CCA instead of CCC, they code for the same amino acid. So it's not a big deal. Okay. While most mutations are not so good for you, once in a while they're beneficial. There's one on the X chromosome that gives women tetrachroma vision, so they can see a lot more colors. Okay, so that was a mutation that Instead of seeing 1 million hues, you can see 100 million. And I think we're going to run out of time, but I think we're done. Yes.